your slides.
Yeah, so good morning, everyone. I'm a research fellow in Ben Snail Group. My background is actually epidemiology and biostatistics, but I'm recently working on whole exome sequencing and whole genome sequencing in the context of psychiatric disorders. What I'm going to talk to you today is about educational attainment, and so I'm moving a little bit out from the diseases and I'm more looking into the general population, and I want to see how these variants, which has been described to be associated with schizophrenia, autism, and other disorders, actually apply in the general healthy population on some cognitive traits. Here in my presentation, you can find my email and my Twitter in case you want to ask any question, both by email or tweet. Okay, so why we want to study educational attainment? Educational attainment is a surprisingly good indicator of life expectancy. As you can see in this graph, women, when they are 25 years old, they have a degree that is less than high school, they have a general life expectancy of 50 years old, which increased to 62 years old. So 12-year more life expectancy in individuals, I guess, very similar to the one that are now attending this conference, which have a graduate degree. So it's naturally not all causative, most is just correlation, but it's a really important indicator of general health status and health attitude. The second point is that educational attainment is highly heritable, and so it's an interesting trait when we want to study the genetic determinant of educational attainment. And finally, educational attainment is highly genetically correlated with IQ. And IQ, it's a kind of a nice phenotype, but it's really difficult to collect on large scale because it's a relatively complex measure to assess. And so while educational attainment is considerably easy to obtain, especially in large epidemiological cohorts. So educational attainment is really an interesting phenotype to study. We also want to study a specific subset of variants, and I'm looking in particular at ultra-rare variants, which is the class of variants that has been previously described by the other speakers. And the reason to do that is that we really need to reduce our searching space as much as possible when we think about association study, because as described by the previous speakers, we don't have enough power to do association study across the entire spectrum of rare variants. And so to reduce the searching space, we want to use information from different sources. For example, we can use population genetic expectation or use genes which are previously associated with diseases or medically relevant quantitative traits. So in this talk, I will focus on a specific subset of variants and gene, which I call disruptive and damaging ultra-rare variants in highly constrained gene. And I will first describe you what these words means and why we want really to focus on this class of variation, why we want to reduce our searching space when we are focusing on rare variants. So for disruptive variants, I mean variants, genetic change that predicted to completely disrupt the function of protein coding gene. So other speakers, they call it loss of function or protein truncating variation. I call it disruptive. We have damaging variants, which are genetic change that result in a different amino acid sequence. I will specifically speak about damaging missense mutation, so our missense mutation which has been candidate to be damaging by a combination of different algorithms. Finally, we look at ultra-rare variants. Ultra-rare variants, I think Giulio spoke about it, are variants that are observed only once in the study. So they are singleton in the study of interest that I'm examining, and they are not inexact. And finally, I look at highly constrained genes, just genes that are sensitive to mutational changes. This is work by Jack, Kathleen, and Mark Daly. So there are genes that across generation accumulate less loss of function variants than what is expected under a neutral model. So to summarize, disruptive and damaging ultra-rare variants in highly constrained gene are variants that are likely to disrupt the function of the gene in genes that are intolerant to this type of mutation. So really bad mutation, which are likely to be associated with diseases under purifying selection. So it seems a reasonable class of variants to study to reduce, as I mentioned, our searching space. And that's been previously studied before. That's been associated with autism, and they've been associated with schizophrenia. So this is the work by Giulio Genovese. You can see that when you look at all the disruptive variants, you see a slightly increase in the frequency of these variants across individuals with schizophrenia compared to controls. But really, you can separate signal from noise when you specifically focus on highly constrained gene versus non-highly constrained genes. Okay, so and I describe a little bit the aim of the study, which is look at educational attainment, which is an important trait in the population, which is a marker of cognition, and we focus on a specific class of genetic variants which has been previously associated with psychiatric and neurodevelopmental disorder and is a candidate to be associated with diseases. So the first research question is, what is the impact of this ultra-rare, disruptive and damaging mutation in highly constrained gene and educational attainment in the general population? And the second question is, what's the contribution of different form and genetic architecture on educational attainment? There's been several studies looking at common variants and looking at copy number variation, CNVs. And so what we want to do is we want to put in the same picture ultra-rare variants, common variants, copy number variation, and see how this different genetic architecture contribute to educational attainment. Okay, so what's our sample size? We put together around 14,000 individuals, mostly from Sweden, Finland, and Estonia. They are enriched for schizophrenic patients, so 36% of these individuals are schizophrenic, but I will show you separate analysis for control, so for individual without schizophrenia, individual with schizophrenia. And we have for 81% of these individuals, we have all exome sequencing, and the remaining is all genome sequencing, high coverage all genome sequencing. This is the outcome that we want to study, this year of education. This has been standardized across these different Northern European countries and put on the US scale for years of education. And what's our predictor of our model is disruptive mutation, damaging mutation, and synonymous mutation. So disrupting and damaging, I previously described them. Synonymous mutation are mutations which are not expected to have impact on gene function, and so they are our negative control. Okay, so I'm going to show you some results. What we basically simply did was to study the association of these three different scores, this burden of disrupting, damaging, a synonymous mutation with year of education, with this outcome. So a simple linear regression that associates these scores with this outcome. Okay, so here on this graph, I'm showing the changes in years of education for one additional of these ultra-rare mutation. And here you have the different studies, and here you have the meta-analysis results. So you can just simply focus on the meta-analysis result if you want. These numbers here are p-value, here are the point estimate and the confidence intervals. When we look at synonymous variants, which is our negative control, as you can see, we don't see an association. It's a nice indication that there's no population stratification or any other bias that affect our observation. When we look at damaging mutation, we start to see a decrease in year of education, and highly significant decrease in year of education. And when we look at disruptive variants, we see an even stronger decrease on years of education. Okay, so it seems to be an association between these burden ultra-rare variants and educational attainment in the general population, a decrease of years of education. But I told you before that I included some schizophrenic patients, and we know that these ultra-rare variants are associated with schizophrenia. So you might think, oh, this result is not unexpected, it's probably mediated by having schizophrenic patient in your data. So when I stratify on individuals with schizophrenia and without schizophrenia, you can see that the effect is the same among schizophrenic cases and controls. So you have the similar effect. Here you can see again the meta-analysis results. It's very similar between cases and control. And we could go even further, and we could link our Swedish data set to registry data, and we exclude everyone that had any type of intellectual disability or neurodevelopmental disorders, and we confirmed that the results were basically the same. So that this signal, this reduction in years of education, is not driven by having some extreme cases of intellectual disability or neurodevelopmental disorders. So just to speak a little bit about the effect sizes, for one additional of these disruptive ultra-rare mutation in highly constrained gene, we observe on average half semester less of education, 2.7 months. This is slightly higher than what is seen for common variants. So there's been GWAS for genome-wide association study for educational attainment, and the top hits is around 2 months less of education. So the effect that we see for these ultra-rare variants is stronger than the one for common variants. We could actually boost our signal when we intersect brain express gene with highly constrained gene. If you remember, we are looking at this impact of these disruptive ultra-rare variants in this specific gene set, which is highly constrained, which is genes that are intolerant to this type of mutation. When we overlay highly brain expressed gene and highly constrained gene, and we have a subset on around 600 genes, we can see that the signals almost double. And it was really the intersection of these two groups that boost the signal, because if we just look at brain expressed gene alone and not intersected them with highly constrained gene, we couldn't see any signal. Okay, so there is an association between this ultra-rare disruptive mutation and educational attainment in the general population in individuals that are healthy and that not have psychiatric or neurodevelopmental disorder. Now, how this specific class of variants play in the picture of genetic architecture of educational attainment? We create score from different sources. We took common variants from this recent published genome-wide association study, which included more than 500,000 individuals, and they look at educational attainments and they identify 7-4 loci, which are associated with educational attainment. We then took these other CMVs, which has been associated with cognition and educational attainments in control. So these are really large, rare pathogenic CMVs, which are also associated with autism and schizophrenia. And finally, we look also at run of homozygosities because this specific class of variation has been associated with educational attainment as well. So what I'm going to show you now is the effect on year of education of this different score. What we did is that we normalized the score, so now the effect size across the score is comparable. And if a score sits on this side of the graph, means that they explain more variants. When it's set on this side, it means that they explain less variant. Okay, so the score that explain less variants are the pathogenic CMVs. It's clearly a single CMV per se, so the few individuals that carry a single CMV, we have a much larger effect size, but by being so rare, the aggregate of the CMV score explain less variant. We have then runoff homozygosities. As you see, all these scores are associated with educational attainment independently to each other. So these are fit in the same model. I mean, the contribution is independent to educational attainment. And then we have our score, and disruptive plus damaging ultra-rare variants. And really, the winner in this competition, who explain more variants on educational, more genetic variants on educational attainment, it's the polygeny score. So clearly, even if the effect size for each of the common variants is smaller, as an aggregate, the polygeny score explain more variants, while the effect of a single ultra-rare variant, the effect size is stronger, but by being rare, they explain less variant. Okay, so now what happens if we look at the interaction between the polygenic score, so built out of common variants, and the ultra-rare variant score? Are the two scores really independent, or they kind of interplay with each other? So what I'm plotting here is the effect of the polygeny score. So I'm basically taking this graph now, this point on the graph, and I'm now splitting it in individuals that do not have ultra-rare variants and individuals that do carry ultra-rare variants. Really, the question here is what the impact of the common variance polygeny score on educational attainment once we stratified individuals who carry these ultra-rare variants and those that do not carry these ultra-rare variants. So as you can see, individuals that do not carry ultra-rare variants, they have a stronger impact of the polygeny score on educational attainment, while individuals that carry some of these ultra-rare variants or pathogenic CMV, which we believe behave in a similar fashion, they have a less stronger impact. So there's clearly an interaction. You can fit a linear model and test for interaction between these two scores, and you will find there is a significant interaction between the ultra-rare variant score and the common variant score. So some conclusion. This burden of ultra-rare disruptive variants, which has been associated with intellectual disability, autism, and schizophrenia, is associated with educational attainment in the general population after we exclude any of these more rare disorder. So as an effect on a continuum of cognition in the population. Common variants, disruptive ultra-rare variants, runoff homozygosity, and pathogenic CMVs, they're all associated with educational attainment, and they're all fitting in this puzzle with different degree of variance experience, so with different degree of importance, but they all fit in the puzzle of the genetic architecture of educational attainment. We found these ultra-rare variants, which is really something new that was not being shown before. They explain more variability than copy number variation of runoff homozygosity. And finally, we found, I think for the first time, when we show an interplay between a rare variant score and a common score. And so we really show that the effect of the polygeny score on educational attainment is stronger amongst those with a lower burden of ultra-rare variants. Okay, so this some conclusion. I want to thank a bunch of collaborators in Boston, in Sweden, in Finland, and in Estonia, which have contributed their exome sequencing data and whole genome sequencing data for this project. I want also to just mention that to process this large amount of exome sequencing data, there is needed for a software that it's able to handle such a complexity of data. And so we were really creating a new software to handle this type of data. The paper is available on the bioarchive on this link if you're interested. Okay, thank you very much.